You're listening to Antiviral Frontiers with your host, Rick Kiesett. In this episode, Emeritus Professor Ruth Itzhaki discusses her extensive research into the link between herpes simplex virus and Alzheimer's disease, and the challenges she faced in publishing her findings.
Welcome to Antiviral Frontiers. I'm Rick Kiesett, your host, and our guest today is Ruth Itzhaki. And Ruth, welcome. And can you tell us a little bit about your background?
Yes, sure. I started off life a long time ago as a physicist, but since then I went via biophysics. I eventually ended up in sort of molecular cell biology - virology, very mixed number of topics really. And I started work on Alzheimer's disease, working first on aluminium, but I gave that up because of technical difficulties which exist with trying to look at aluminium. In about 1989, I think it was, yes. And then I started looking for evidence of a viral role in Alzheimer's disease. There had been a suggestion in about 1982 by an American neurologist that herpes simplex virus might be involved in Alzheimer's disease. And in fact, in 1986, a few years earlier, I and some colleagues had done some work looking for evidence of virus because we had some samples that were very unusual and very interesting, which one wouldn't normally get, and that's people who had been immunosuppressed. And to cut a long story short, those who had been immunosuppressed and who were seropositive for herpes virus, in other words, herpes simplex virus was present in their bodies, they showed evidence by in situ hybridization, which was the technique used then, which is quite insensitive compared to PCR. Anyway, they showed evidence of viruses in brain, quite strong signal. But those who had not been immunosuppressed or who weren't seropositive for herpes did not show anything. And I think that the reason was simply that the technique was not sensitive enough to detect the virus at its probably very low level in brain. But when immunosuppressed, the virus was able to reactivate from latency, which we think is how it exists in brain, to productive infection. And then, of course, there were far more copies of the virus, which meant it was detectable by this rather insensitive technique. So that was a very interesting study, but we didn't follow that up for various reasons. But I decided to look in about 1989 for evidence of the virus by PCR, which had just started to come in during the 1980s, and that hadn't ever been used before to look for virus. So we decided to look for it, and we found evidence of the virus, very interestingly, in both Alzheimer's disease patients and age-matched normals, which puzzled us quite a bit at first, but I'll continue with that in a minute. But we had extraordinary trouble in getting that work published, which is something that's occurred for the subsequent 25 or more years. People just not believing it and saying we had contamination, which we took very great care to avoid, having everything sterile and so on. And so anyway, to continue why we thought that virus was present, or why we'd found virus in both, we considered that it probably was caused by the fact that another factor was needed to act together with the virus, a genetic factor of the host. And I did a very speculative experiment, or my student did, looking at herpes simplex, HSV1, whether people with cold sores were virus positive or not. I should explain, perhaps for those who don't know, that about between 25 and 40% of people who suffer from cold, who are infected with herpes simplex virus, HSV1, show cold sores. The remaining people, the other 75 to 60%, do not have cold sores. So it's obviously again, possibly to do something to do with the genetic factor. And what we found was that a genetic factor which was known at the time to be a risk for Alzheimer's disease, the ApoE type 4 allele, was present in a high proportion of the people who got cold sores, but only in a low proportion in those who didn't get it. And this was the case also we found for people who had herpes virus in brain and got Alzheimer's disease, but a much lower proportion had an E4 allele in people who had the virus but didn't get Alzheimer's disease. So what we reckoned and what we published in 1997, again with huge trouble getting it published, was that the combination of carriage of an ApoE4 allele together with virus presence in brain conferred a strong risk of Alzheimer's disease. And perhaps I could point out now something that a lot of people hadn't really been aware of, an astonishing number actually, that sometimes, very often, if not always, susceptibility to an infection and also the degree of severity of the infection for those who are infected depends probably on genetic factors of the host. And so you can be infected, but not affected. And this is something that, say, a lot of people have great trouble in understanding. Yet it's true of, very true, for example, of tuberculosis, where only about 1 in 10 people, 10% of people who are infected with TB actually, with the TB bacterium, actually develop TB. So it obviously depends on factors other than just the infectious agent. I think I should stop there, but I could go on for a long time, and I better not.
Yeah, that's very interesting. And what about amyloids and abnormal tau and so on? Did you find association with that as well?
You're saying, what about amyloid? Is that right?
Yeah.
Yes. Well, we think it's not a cause, although it was postulated to be one for a long time, but we do think obviously it's important, a symptom of the disease, very important, and perhaps involved in the process, almost certainly involved in the processes that lead to the disease. So we think it's a symptom, not a cause. So I think it's we
like a repair, some repair mechanism, or is it like a repair mechanism, do you think?
Well, yes, that's
or
another
isolating
thing.
the virus some way?
Yes, that's what I was going to say. We found that when the virus infected cells in culture, amyloid was produced, and this was very exciting. And again, I had awful trouble publishing that. People just didn't believe it. We also wondered, actually, way back in about 2006 or so, or 2005, that the amyloid might possibly be an antimicrobial factor. And we did some experiments with bought amyloid to try to find out about that. And that was really early on that we did this. But the quality of the amyloid was such that it proved fatal in many cases to the cell, killed the cells. And so our results, although it did show protection, weren't really valid, which was a great pity, because subsequently it was found a few years later that in fact amyloid does have antimicrobial properties. So what we think is happening is that, as we showed, infection with the virus causes production of amyloid, also by the way of abnormally phosphorylated tau, and probably it's there to combat or try to combat the infection, but at a sufficiently high level, which it perhaps eventually becomes because it accumulates, it probably becomes toxic. This isn't actually often stated, but we think this is what is actually happening.
I see. Interesting. So here at Keimer Med, we're working on antiviral drugs. And can you tell us a little bit about the work you did with antivirals and your research?
Well, we infected cells with herpes HSV1 and looked at the effect of several antivirals using as criteria of damage production of amyloid and production of abnormally phosphorylated tau. And we showed very clearly that, in particular, acyclovir was very effective and several other antivirals as well, very effective in reducing the levels of these, particularly of amyloid, or particularly of tau, which went down to a very, very low level at a fairly low dose of antivirals, but also a considerable reduction in the amount of amyloid. So it seems to be that I think it really, antivirals are very effective. I should mention now that the recent antiviral trial, which was carried out at Columbia University, very unfortunately did not seem to work. And I think there are possibly two reasons for that. And the first one is that the people who were dosed with valacyclovir, which breaks down in the body to give acyclovir, probably by then the virus had done most of its damage. They were at a stage in the disease where it was mild cognitive impairment or full Alzheimer's. And we think what should have been done was that people should be dosed very much earlier, although the problem here is detection of the early stage. And the other one is, I am not a clinician, and I wouldn't like to pass an opinion on this, but somebody I know, a colleague, thinks that they should have been given a higher dose of acyclovir. So it's one or other or both of those, and me hoping very much that some trial might be carried out much earlier. In fact, in about 2011, something around that time, I suggested in one of my papers that people who reach middle age, late middle age, those who are herpes HSV1 positive, seropositive, should take antivirals like valacyclovir regularly in the hope that it'll act as a prophylactic. But of course, nobody's able just to do that work because there's no inadequate preliminary data to support it. So it's something that I think, like statins, should be used, but where the money will come from in the absence of any concrete evidence, I don't know. But the problem is, how do you have to look for indications that the people have the virus and yet haven't shown signs of Alzheimer's.
I see. Very interesting. Are there other viruses that you also found to be associated with Alzheimer's, or is it just HSV1?
Yeah, well, there's certainly now people are thinking that maybe varicella virus will the one that causes chickenpox and shingles, VZV, might be involved, because there have been these very interesting experiments, which I think did one of the first studies on shingles vaccine, which we published in 2021, showing that treatment with Zostavax in that case, which was the main anti-shingles vaccine used at that time, had a fairly large effect on subsequent risk of Alzheimer's disease.
Oh, really?
And since then, there have been about 5 other studies, of which probably the most significant was it took account of various possible confounding factors. The one by Pascal Geltzelser, which was published in either Nature or Nature Medicine, I can't remember which showed very clearly, as did all of the papers, that the risk was diminished if for those who take who have a Zostavax or Shingrix vaccination.
Is the reason for that known? Is it just immune system? Is the reason known? Is it immune system stimulation?
No, it isn't known. Pascal also gave several explanations of it, one of which was the one that we'd suggested in 2021, which was that we think that when people are infected or have various other types of injury, head injury, and so on, various things that are known to be risk factors for Alzheimer's, that an injury, which is supported by quite a lot of data in life, that injury from infection or other types of injury causes reactivation of the viral latent herpes HSV1 in brain, and then that causes subsequent damage. And if this happens recurrently, then eventually it leads to Alzheimer's disease. And we showed evidence of this we published a paper showing that addition of varicella zoster virus to a 3D cell brain model, this is in collaboration with Dana Cairns and David Kaplan in Tufts University. Using their 3D model, we showed that in cells which are quiescently infected, probably latently infected with the virus, addition of VZV and also of other viruses, which we haven't yet published. We've published only the VZV work, cause very striking reactivation of the HSV1. I should say this was done after a study they published in 2020, showing very important study from my point of view, and those who support a viral involvement, showing that in their 3D culture, infection with HSV1 causes pretty much all the main symptoms of Alzheimer's disease. So this was very strong support for the viral hypothesis. But in this case, it was treating cells with an infection, VZV, and seeing what that does to quiescent, the latently infected virus. So I should say there's also strong evidence from work by various people, various groups, showing that infections can cumulatively cause a risk of Alzheimer's disease. I mean, it was known to be a risk factor. In fact, some infections were known, but this was sort of cumulative effects of infections together can lead to Alzheimer's. And then subsequently, we did a similar sort of work using head injury or an impact on the 3D and 2D cultures that we used, mimicking head injury. And they showed the same thing, that this type of damage causes reactivation of the virus. I'm not saying it doesn't cause other types of damage, which matter, but I think it's very significant because there's quite a lot of evidence that head injury does lead to, in some cases, at least to Alzheimer's disease eventually, and boxers and footballers. So I think it was quite significant. Also, strongly suggests that risk of infection should be looked at. People should there should be efforts to reduce the risk of infection. There's another strong risk factor.
Does that suggest that other types of infection, say bacterial infection in the brain or spirochetes or those sorts of things are also potential triggers? Are they also potential triggers, those other types of infections?
I think that there's reasonably good evidence, not as much with HSV1 as it happens, but that's only incidental, simply not been as much work done on these others. Certain bacteria, including the oral one, and others, chlamydia pneumoniae, for example, and also, for that matter, various fungi could be a cause of the disease. Whether they're acting separately or whether they act in combination or in combination with HSV1 isn't known. But I think the fact that infections do cause eventually accumulation of them, cause Alzheimer's, I think is quite significant. And really the question is actually proving causality rather than just an association, which our critics, who actually never produce any scientific arguments, their main point really is that we can't prove causality. But causality in a human disease, of course, is very hard to prove because you can't go and infect people and then see if they develop it.
Right.
So it's rather a spurious argument in a way, or superficial one.
Yeah. Is it known whether the latent form of herpes is damaging? Is it only when it transitions to lytic that it damages, or is it also causing havoc in latent state, or is that known?
Yeah, well, it's a good question. It might be. Not much really is known about what happens during latency. There's certainly very limited transcription. Nobody, I think, has done any experiments showing is damaging, and one might expect much more damage, if there is any at all in it, much more once it reactivates, simply because of the huge increase in the number of viruses replicating and then thereby doing damage. So it's a very hard question to answer, because there are not very many, or if any, really suitable in vitro models.
Right.
I should say, by the way, there's other work that suggests that supports the, of course, reactivation of HSV1 hypothesis. In fact, there are now something like 600 papers, nearly every single one of which supports the viral role, HSV1 in particular. And they're using a great variety of approaches and methods, which makes it reassuring.
Right. And what about the different parts of the brain that are affected by the virus versus Alzheimer's? Are they the same, or are they?
That's again something we don't really know, except from their effects. I mean, obviously there's a big effect on memory, big effect on things like activities of daily living, but what is actually going on in the brain in life isn't known, because one of the main problems at the moment is you can't detect the virus in brain, probably because the level is so very low. So you can't follow what it's doing. And if somebody were, if that becomes possible, that would be a big breakthrough, because of course you can't even prove that it's apart from PCR or other methods, you can't be absolutely sure that it's there. It might be the virus might possibly be acting from, say, the trigeminal ganglia and the peripheral nervous system. It might reactivate temporarily. This has been suggested, go to the brain and then stop. But we don't really know much about this behavior.
Interesting. And what about the pathway into the brain? Is that known? Is it go through the dental nerves or blood-brain barrier or olfactory, or is that it seems only a limited number of ways, right?
Sure. Well, blood-brain barrier probably isn't relevant, because the virus is rarely present in blood. But the olfactory route is certainly a possibility, or reactivation traveling along axons to the brain from the trigeminal ganglia. And there really isn't, again, adequate evidence of whether both of those are acting or whether only one of them is. It's extremely hard, to follow. Say, in life, you simply can't do it, really, at the moment. So,
what about in like cerebral spinal fluid or something like that?
Well, we did some work with CSF, and that was actually, at the time, seemed a very nice proof of the presence of the virus in brain. We detected antibodies to the virus in brain. They are, the virus itself, if it's in the CSF, very short-lived. You can detect it for only about a week or two after herpes simplex encephalitis, which of course is a very severe acute form of herpes infection, where it's definitely the virus that's causing the encephalitis. And the virus, is there for a week or two only, but you can detect antibodies to it for up to several years after, although there are cases probably where it's not present, and obviously various reasons for that. We detected it in rough similar proportion of brains as those proportions we found by PCR, which was very exciting. But I've been trying to look into this, but at least one person has tried to reproduce these results and not found not got the same results, and we don't know what the reason for that is, which is a great pity. But I should say, by the way, we did also in situ PCR, which is a very difficult technique, but indicates where the virus is, and that showed the presence of the virus in brain too from tissue sections. So that was rather reassuring. But the business of CSF is quite puzzling, and I can't explain that at the moment, unfortunately.
I see. Does any of your work suggest a potential treatment or prevention? You mentioned, the idea of taking cyclovir. I guess when you're past a certain age, are there other things like that elderly people might consider as preventative or treatment oriented based on
Well, I
your
think
research?
there's a lot of interest in the possibility of a vaccine.
Mm-hmm.
Because of the shingles work mainly,
Yeah.
vaccine against HSV1, but so far that hasn't been produced. But I think that's a very interesting approach, as well as I hope that antiviral treatment might be used from middle age onwards for those who are seropositive for HSV1, and particularly those, of course, who carry an ApoE4 allele.
Yeah.
As I mentioned before, we think this might well have an effect because that's probably in middle age when the might well be it looked from our preliminary results with CSF when the virus gets to the brain.
Have you looked at how the VZV vaccine helps reduce the risk of Alzheimer's?
Well,
Is
no,
it
it isn't.
adaptive immune system reaction, or is it the adjuvants in the vaccine, for example? Is that known?
Yeah, that's an interesting question, of course, again, because BCG, which is probably what you're alluding to, has been found to have these side effects. Sorry, there's a term for it, I can't remember what it is, where it seems to affect or reduce the risk of a number of diseases besides those TB, which is supposed to act. Very, very interesting, and so it might be the adjuvant that's working. But at the moment, there's no vaccine against HSV1 at the moment, unfortunately, so that needs to be discovered. But the action what we think is happening is when the virus replicates, it eventually it causes inflammation in the brain, and that causes damage. So it's sort of indirect in a way, as well as probably direct viral damage. But it's well known that inflammation not only reactivates the virus, but it also causes inflammation once it has reactivated. So it's a vicious circle, really.
I see. Does that suggest anti-inflammatories would be helpful as a
Yes.
treatment?
Well, anti-inflammatories as such, so I think people have tried statins and they're a bit ambiguous, the results. So it's probable that one needs something which actually attacks the virus as such. No, it's a great pity that there's no, at the moment, the only possible mechanism to stop it happening might well be antivirals. And for that, as I said, we have no supporting data, unfortunately, not yet anyway.
And why do you think it is that HSV1 in particular is the culprit in the brain, as opposed to why don't other viruses that reach the brain also cause Alzheimer's? Or maybe they do. I guess that's a question.
Well, the reason that it was suggested in the first place was by a American neurologist, Melvin Ball. And he suggested in 1982, when he pointed out in a paper, theoretical paper, that the regions of the brain that are affected most are those that are affected most in Alzheimer's disease. And other arguments in support of its being HSV1 is the fact that it can be dormant, which all herpes viruses have this characteristic. So it's possible that other viruses might be involved, but there's very inadequate data suggesting that. For example, we looked for varicella zoster virus DNA in brain. We didn't find it, but that was in 1998 or something, I think. And techniques of PCR were probably much less sensitive than they are now. So that might well be something we'll probably repeat. As to other viruses, well, VZV might be involved, but as we didn't detect it in brain, it might or might not be. There's even less evidence for other viruses like HSV2 or CMV. So although they all have this characteristic of being able to be latent, which is very... Another argument in favor of its being HSV1 is the fact that infection with HSV1 is so common.
Sure. What about the HHV family?
Well, HSV6
6 or 7.
HSV6. Yes, we've been doing some experiments with that, which we hope to publish soon. It does cause reactivation, by the way, in the way that VZV does in the 3D cell culture. But we couldn't find evidence that it actually acts, which we'd previously suggested, having detected it in brain. I mean, it's known to be, it can occur in brain, definitely. And we didn't find evidence that it had any additional effect, any synergistic effect with HSV1, or even a definite effect on its own. So I think that's quite uncertain at the moment.
I see. Does the viral load increase with age?
Sorry, is viral load what? Sorry.
Does the viral load increase with age in the brain? Is that known?
Well, we could only do that
like
really
immune decline, immune system decline, or something.
Sorry. Well, we have no evidence of the actual load. We didn't have the money to do any quantitative PCR, which was coming in during the time when we were looking for the virus. We were incredibly short of money all the time because of the great hostility to this work, which meant we couldn't get grants and we couldn't get publications. So yeah, had a pretty tough time for about 25 years, I should say. We were the only people in the world working on it for quite a few years, and then luckily other people joined in. And of course, in the last 5 years, there have been a lot more people joining in, which is very good news. But no, well, I think that's really my answer.
Yeah, interesting. Do you think this concept gaining increased acceptance now? Is there? You say there's 600 papers. Is there? Is it gaining increased acceptance? The HSV1?
Yes, I think it is. But there are still some very influential people in the field who just ignore it or disparage it very virulently, actually. And they're quite influential people. But as I mentioned before briefly, they don't really have any proper scientific arguments against it. I mean, all they can say is you're proving association, but you're not proving causality. I think that's rather inadequate argument with so many different approaches suggesting that HSV1 is involved.
Yeah. In your experiments with amyloid, is there some understanding now of how HSV1 causes amyloid formation?
Just trying to think of any detail known about it. We found it affected one of the enzymes involved with producing amyloid, but as far as I know, nobody's done anything in more detail about that or anything else. So really not much is known what is happening. But I mean, it is consistent with the fact that an antimicrobial agent, that amyloid is an antimicrobial agent, trying to combat the virus presence.
How does that work? How does beta amyloid act as a antimicrobial peptide?
I'm afraid I can't answer that one. I don't know. I'm not sure how well it's known, but I don't know it.
Yeah. Fair enough. Where is your research headed now?
Really, at the moment, interested in. I've been doing some epidemiological work, as I mentioned, with particularly with Shingrix. Well, sorry, with VZV. Really, the work is now turning a bit towards looking into VZV and how it might act. I think the other very interesting thing that needs to be done is to see if one could find out how other infectious agents act, whether they act together or whether they act individually, or whether they act with HSV1. And I think this is something that's very important that should be done, because obviously it would determine what type of treatment you use. So
sure.
I'm hoping that, but I'm not in a position to be able to do that. So
yeah,
concentrating on the other two aspects.
Right. Very interesting. Where would you like to see this research with Alzheimer's go from here forward? Based on the work you've done and where it's been.
I suppose the obvious thing is prevention, which is really much, much better than treatment, which is uncertain anyway, because obviously the numbers of people living longer is increasing, and that means there'll be more people Well, I think the numbers are projected to increase greatly, but in fact, to some extent, there's more interest in affecting the risk by changing your behavior by what you eat and exercising and so on. So there are various steps being taken to ameliorate the situation. But obviously, prevention, if that can be done, will be very good, because if people are going to live longer, they should hopefully have a decent life when they're alive, for the last few years, rather than suffering all sorts of ailments, which make living sometimes quite a misery.
Indeed. Well, it's been great talking with you, and thank you so much for your time.
Pleasure. Well, I enjoyed it. Thank you very much. I liked your questions.
We hope you enjoyed this episode of Antiviral Frontiers. To hear more from the fascinating world of viruses, biotech, and medicine, please visit our website or find us on Spotify and YouTube and follow us to get future episodes.